Welcome to the Folate Microbiologic Assay training video. The purpose of this video is to demonstrate key steps of the Folate Microbiologic Assay. When women of reproductive age have low blood folate concentrations, their offspring have an increased risk of developing neural tube birth defects. Surveys that determine folate status help countries assess the extent of these folate deficiencies. Then, they can determine if interventions such as fortification or supplementation are needed. Knowing the optimal blood folate concentration needed for women of reproductive age to reduce the risk of neural tube birth defects is key to interpreting the data. A recent guideline published by the World Health Organization provides this information and recommends the use of the microbiologic assay to assess folate status in population surveys. Different folate assays produce different results. This makes it difficult to interpret data from multiple sources or use the same cutoff value to assess folate deficiency or insufficiency. The Folate Microbiologic Assay is the assay of choice for population surveys because it responds similarly to all biologically active forms of folate, requires a small sample volume, and does not use expensive supplies or instrumentation, making it particularly suitable for low-resource settings. Furthermore, recent findings show that when different laboratories use the same critical reagents, such as the microorganism and calibrator, in the microbiologic assay, the comparability of their results improves. Before we move on, please remember to consider all biological specimens to be potentially infectious. Therefore, it's critical that you observe universal precautions, wear appropriate protective personal equipment, and follow good laboratory practices when handling chemicals. Let's first review the principle of the Folate Microbiologic Assay. We first add the microorganism Lactobacillus rhamnosus to growth medium containing all the nutrients the microorganism needs to grow, except for folate. Step 1. Then, the inoculated growth medium and diluted serum or whole blood lysate are added to 96 well plates, steps 2 and 3. We incubate the plates for 42 hours at 37 degrees Celsius, step 4. We then assess the folate concentration by measuring the turbidity of the inoculated medium at 590 nanometers in a microplate reader, step 5. The growth of the microorganism is proportional to the amount of folate present in the sample. To calibrate the assay, we use 5-methyl tetrahydrofolic acid, the main folate form in serum and in most whole blood samples. This training video consists of 7 parts, as shown here. Prepare reagents. Set up and dilute samples. Prepare 96 well plates and add reagents. Dilute calibrator and add to calibrator plate. Add diluted samples to sample plates. Seal and incubate plates for 42 hours at 37 degrees Celsius. Read plates at 590 nanometers. To facilitate the setup and maintenance of the microbiologic assay in low-resource public health laboratories, CDC has assembled a frozen assay kit containing the following components: microorganism, calibrator stock solution, 3 reagents needed for the preparation of the assay medium: ascorbic acid, chloramphenicol, and manganese sulfate, and 2 levels of quality control materials. Not provided with the kit are the following items: growth medium, tween 80, sodium ascorbate, and deionized water. 2 reagents need to be prepared freshly for each run. First, the sodium ascorbate. To prepare a 0.5% solution, add 2.5 grams of sodium ascorbate to a 500 milliliter beaker and dissolve with 500 milliliters of deionized water. Next, the medium. To prepare 200 milliliters of medium, add 14.1 grams of folic acid KCI medium to a 500 milliliter beaker. Then add 200 milliliters of deionized water. Heat to boil while stirring and keep boiling for 3 minutes. Cool down to room temperature. Add 1 vial each of ascorbic acid, chloramphenicol, and manganese sulfate solution. Add 60 microliters of tween 80. Mix well for a few minutes. Thaw a vial of frozen microorganism and add 700 microliters of the inoculum to the medium. Stir gently. Let's review the major equipment and supplies needed for the Folate Microbiologic Assay. 37 degrees Celsius incubator, microplate reader, 360-degree rotator, microplate heat sealer, various pipettes and corresponding pipette tips, vortexer, stirring hot plate, general laboratory equipment such as a balance, freezer, and refrigerator, 96-well microplates, sealing membrane, and general laboratory supplies such as glassware, racks, and various plastic and glass vials. Keep a run sheet to make note of medium lot number, calibrator lot number, sample dilution factor, or any other specific information. To minimize errors during sample dilution, use a parallel setup for sample vials coming from the collection site and for the glass tubes that contain the diluted samples. Add QC and blank samples in a 12x4 rack. While samples are thawing, label glass tubes and arrange them in a separate 12x6 rack. One 12x6 rack holds 48 samples, which fit into 2 96-well plates. An experienced analyst can comfortably prepare 5 96-well plates per run, 1 calibrator plate, number 1, and 4 sample plates, number 2 through 5. In a run with 5 plates, each sample plate contains 1 QC sample, 22 unknown samples, and 1 blank sample used as a negative control. Thus, each run consists of 2 low and 2 high QC samples, 88 unknown samples, and 4 blanks. Use an automated repeater pipette with a 25 milliliter tip to add sodium ascorbate solution to the glass tubes. For a roughly 1 to 100 dilution of serum samples, use 1,475 microliters of sodium ascorbate. For a roughly 1 to 140 dilution of whole blood lysate samples, use 2,075 microliters of sodium ascorbate. Use a single-channel manual pipette, 10 to 100 microliter range, to transfer 15 microliters of well-mixed serum or whole blood lysate sample into the glass tube containing the appropriate amount of sodium ascorbate. Ensure that there are no air bubbles in the pipette tip. Cap each tube. Keep the original sample vials in a refrigerator until later when you scan the sample IDs into the microplate reader software. Then return the vials to the freezer for future analysis if needed. Label each plate on the front with the date and analyst initial. Write the plate number on the upper left corner and on the front. Plate number 1 is used for the calibrator, and subsequent plates are used for samples. Write the different volumes of sodium ascorbate and calibrator for each column on the plate. To help with correct transfer of diluted samples into the plate, divide the plate into columns and rows as shown here. Each sample will occupy 4 wells. Use a marker pen to label the sample number on the sample plates. The first 2 reagents that need to be added to the 96-well plates are the growth medium and the sodium ascorbate solution. Use a 12-channel electronic pipette, 1,250 microliters, with the repetitive pipetting function and volume set to 200 microliters to add 200 microliters of growth medium to all plates. To add sodium ascorbate to plate number 1, the calibrator plate, use a smaller 12-channel electronic pipette, 300 microliters, with the single pipetting function. Transfer appropriate volumes into each column as shown here. Next, use the same 12-channel electronic pipette, but now with the repetitive pipetting function and volume set to 50 microliters to add sodium ascorbate into plates number 2 through 5, sample plates. But only add the solution to the odd number columns. Cover all plates with foil to protect them from dust and light. Next, work on a different bench to prepare your calibrator working solutions to avoid potential contamination of the bench where you set up the run. We have 4 pipettes that we use exclusively for this purpose. They are set to 50, 100, 250, and 400 microliters. Thaw 1 vial of frozen calibrator stock and mix it well. To prepare a 1 to 5 diluted stock solution, add 400 microliters sodium ascorbate to a 2 milliliter cryo vial. Then, add 100 microliters calibrator stock solution and mix well. Label 2 50 milliliter grade A volumetric flasks as level 1 and level 2. Add approximately 45 milliliters of sodium ascorbate to each flask. Transfer 50 microliters of diluted stock solution to the level 1 flask. And 250 microliters of diluted stock solution to the level 2 flask. Using a plastic transfer pipette that has a fine tip, make up with sodium ascorbate to the 50 milliliter volume mark. Be sure that the bottom of the fluid meniscus touches the calibration line of the volumetric flask at eye level. This is a very critical step to achieve accurate results. Slowly invert the volumetric flasks 20 times to thoroughly mix the calibrator working solutions. Now you can move back to your sample preparation area. Change gloves to prevent accidental folate contamination. Pour the level 1 and level 2 working solutions into 2 labeled basins. To add calibrator working solutions to plate number 1, use the smaller 12-channel electronic pipette, 300 microliters, with the single pipetting function, and transfer appropriate volumes of the working solutions to to plate number 1, as shown here. Gently hand seal the calibrator plate with a sealing membrane and set it aside, as well as the basins containing the working solutions. Change gloves to prevent accidental folate contamination of the sample plates in the next step. To transfer an aliquot of the diluted samples to the sample plates, use a single-channel electronic pipette with the repetitive pipetting function and volume set to 50 microliters. It is very important that you thoroughly vortex mix each diluted sample prior to taking an aliquot. Add diluted sample into 4 wells each of 2 × 50 microliters and 2 × 100 microliters. Pipette 50 microliters twice to get 100 microliters. The full plate layout is shown here. After finishing with one plate, gently hand seal the plate with a sealing membrane. Preset the plate sealer to 130 degrees Celsius. Once the temperature is stable, place a piece of aluminum foil on top of the sealing membrane to protect the samples from heat. Place the plate on the holder and apply heat for 5 seconds. Remove the foil membrane and immediately place an ice pack on top of the plate for a few seconds to cool down the membrane. Apply additional manual sealing to prevent leakage and thus improve precision. Use a flat object to press the sealing membrane down and to seal edges and corners. Now the plates are ready for incubation. Stack the plates and sandwich them in between 2 empty plates. Make sure the temperature in the incubator is within ± 2 degrees Celsius of 37 degrees Celsius, and that the fan works to distribute the heat evenly. Place the plates in the center of the incubator. Keep the plates incubated for approximately 42 hours. Remember to time the start and end of this process carefully so you aren't forced to remove the plates in the middle of the night. There are different microplate readers available. The reader you use must allow you to 1. Use a wavelength of 590 nanometers. 2. Set up an experiment with multiple plates, because you'll use the calibration curve from plate number 1 to calculate results from the sample plates. And 3. Choose the correct curve fit to calculate results, which is the polynomial third-degree curve. We use the Biotech Power Wave Reader with Gen 5 software to collect data. For more flexibility with the size of your run, either fewer or more plates, you can generate different experiment templates according to the number of plates used in the run. Open the experiment template with the number of plates corresponding to your run and save it with a new name, such as today's date. Scan only the sample IDs for the unknown samples, not including the QC samples and blanks, into Notepad. Then upload the file into the experiment you created. Make sure correct dilution factors are used. If irregular dilutions are used in the run, edit the dilution factors accordingly. Add bleach to the leftover folate assay medium containing the microorganism for a final concentration of approximately 10% and let it sit for 30 minutes to inactivate the microorganism. Place disposable plastic, glass, and paper items, pipette tips, vials, glass tubes, gloves, microplates, disposable bench liners, etc., that may have come into contact with serum or blood into a biohazard autoclave bag and keep the bags in appropriate containers until they are autoclaved and properly disposed. Discard the leftover medium that now has a brown color into the drain. The leftover sodium ascorbate can also be discarded into the drain. Beakers are washed with deionized water, and work surfaces are wiped with 10% bleach solution after the daily work is completed. After approximately 42 hours of incubation, remove the plates from the incubator and place them into an orbital mixer. It takes about 30 minutes for plates to cool down to room temperature and to thoroughly mix. Open the corresponding experiment created 2 days ago. It is important to strictly limit the time from stopping the mixing to removing the sample membrane and reading the plate to 1, maximum 1.5 minutes. During this minute, open the sealing membrane, wipe the plate bottom with a tissue in case there are any particles or dust, and place the plate into the plate reader. To remove any microbubbles inside the wells, gently fan the air above the plate and check for any floating particles inside the wells. Read the plates one by one in exactly the same way. Save the results after each plate reading. Handle all plates consistently. Review the calibration curve and QC results, and the imprecision of the 4 replicate results for the unknown samples following the data review SOP. Generate power export results and save the file. Remember to discard the plates into a biohazard autoclave bag. This concludes our training video for the Folate Microbiologic Assay. It is important to remember that this assay is very sensitive at detecting trace levels of folate, and therefore you need to work carefully to prevent folate contamination. Use dedicated pipettes, glassware, and workbench to dilute the highly concentrated calibrator, and, if possible, a separate sink to clean up your reusable glassware. Also remember to regularly decontaminate the bench surface and pipettes used for sample preparation. We hope that you enjoyed this video and that it will help you achieve and maintain proficiency with this important assay.